So now we're going to start Unit 7, Lecture 2, where we're going to discuss all the minerals that are involved in metabolism. Now, the first mineral we're going to talk about is iodine. So iodine is critical for the formation of thyroid-stimulating hormone and thyroid in general. And the thyroid, if you remember back a few units ago, is really what's maintaining your basal metabolic rate. So if you don't have adequate iodine within your diet, then you're going to end up having a reduced thyroid hormone, therefore a lower basal metabolic rate. And many people are actually plagued with hypothyroidism. Some are plagued with hyperthyroidism, but it's very easy to pick out someone that is hypothyroidism because they just move so slow and have no energy. But so specifically with iodine, it is readily absorbed from the GI tract, with the majority of it is absorbed throughout the small intestine. And it can be absorbed within the rumen of ruminants, so about 70 to 80% of the iodine in ruminants is absorbed in the rumen. In addition, when you have excess iodine, it's primarily excreted in the urine. You have some that you lose with sweat, and that's during severe cases of heat stress. And as I said, the main function of iodine, it is as a component of the thyroid hormone. So iodine is incorporated into the tyrosine residues of the thyroid globulin. And that's how it's stored. In about 90% of the body, iodine is stored as thioglobulin. And then you have the thioglobulin, so the stored form of iodine, this protein, is incorporated into tetraiodothiamine, or T4. So if you've ever had your thyroid tested, they test for T4 and T3. So T3 is triiodothiamine, and those are the active forms of thyroxine. So those are the ones that are actually maintaining your basal metabolic rate. So here we have a diagram of how our thyroxine is actually synthesized. So we have our thyroid cell here, and all of this has to occur within the thyroid cells or the thyroid gland because the thyroid is the only cells that are able to oxidize iodine to its higher valence level. So that's why all the other tissues, they're unable to do this. Only thyroid cells can do this. So we have down here our iodine enters in to the cell using iodine transporter. I'd like to note that it uses sodium potassium ATPase, so this is active transport. So we need to use energy to get iodine within the cell. And once it's within the cell, we then transport that iodine out into the follicular space within the thyroid gland. And it's at this point in time in which we oxidize our iodine. It's now charged, and we're able to bind that to our thioglobulin. And so the MIT is monoiodo thioglobulin and the diiodo thioglobulin. And you can see, so this is made all within the colloidal space, the follicular space within the thyroid gland. And then once we have our thioglobulin made, it is actually phagocytosed or pinocytosed back into the cells. And we have this hydrolysis that occurs, and this is where we're getting the formation of our T3 and T4. And then they're able to go back into the extracellular space, and this is the main way in which we are transporting our iodine, which is our thyroxine. Now, T4 is thought of as the pro form. So once it gets to the cells, the target tissues, then the T4 will be converted to T3, and T3 is actually what's going to be binding the thyroid hormone receptor to transcribe genes. Now, the primary deficiency that you're going to encounter when you are deficient in iodine is actually goiter. So this actually looks like a big softball on the side of the neck of an individual that is iodine deficient. So we have thyroid stimulating hormones. So when we have a need for thyroid, our thyroid stimulating hormone is secreted from the pituitary gland. And when we are deficient in iodine, our thyroid hormones decrease, and we end up releasing more of this thyroid stimulating gland, causing the gland to enlarge. We also have cretinism, and this is due to a lack of thyroxine. So this occurs either due to secondary iodine deficiency or congenital disease. It causes retardation in growth. And both conditions can be caused by consumptions of goitrogens, primarily plants. So these are going to be binding iodine. So cabbage has a lot of these antagonists, iodine antagonists. So they bind iodine, limiting its bioavailability, so the animal's unable to absorb it, and then that results in the formation of goiters. Now, goiters were actually pretty common even in the U.S. Currently, we don't see a lot of goiters because we have iodized salt. So back in the mid-40s, when it was discovered that iodine is the cause of the goiters, and many Americans, even at that time, used a lot of salt as a seasoning. Salt is put in a lot of food, so the government came up with the idea, well, if we incorporate iodine into the salt, which is pretty commonly used in all cultures, then we'll be able to reduce the amount of goiters, and that's basically what happens. So in the U.S. especially, we have pretty much eliminated all cases of iodine deficiency and the formation of goiters. And if you have a goiter and you end up taking supplemental iodine, the thyroid gland won't ever shrink back down. So once you have this hypertrophy of the thyroid gland, it won't ever shrink back down. So typically at those points in times, individuals that have those, they end up having them surgically removed, and then they have to take supplemental or the thyroxine as a drug on a daily basis to maintain their basal metabolic rate. So the physiology of the formation of the goiter, it all starts with our circulating level of our thyroxine, so our T3 and our T4. So as we have lower levels of our T3, so low levels of T3 and T4 are going to have a positive effect on our hypothalamus. So that our hypothalamus is going to release thyroid releasing hormone. This has an effect on the pituitary gland right here. And our pituitary gland, as I mentioned before, is what is releasing our thyroid stimulating hormone. So this thyroid stimulating hormone is going to affect the thyroid gland, and in a normal situation, that's going to cause the thyroid gland to synthesize greater amounts of thyroxine. But if we have lack of iodine, we're not going to be able to form our T3 and T4, as you just saw previously. So therefore, we're going to have this constant loop, and then this is going to get much bigger, and you're going to end up looking like you have a big baseball in your throat. So your thyroid gland is found within your neck, and so here is a normal individual. You can see over here, normal size thyroid. As we have the lack of iodine, or even there's some autoimmune diseases that will reduce the amount of thyroid that your body is making, so you get hypothyroidism. Typically with hypothyroidism, you're not going to get an enlarged thyroid gland because your body's attacking the thyroid. So during our iodine deficiency, our thyroid is going to get much larger, and that's what's going to cause a big ball in your neck. So here's an individual that has goiter. So you can see here this big bulge in this individual's neck. So that is an indication of iodine deficiency. So that's the goiter that's formed. The thyroid gland becomes much bigger, hypertrophies, and that's to compensate, trying to make more thyroxine, but it's unable to do it because of the lack of iodine. So here we have a picture of iodine deficiency. Again, the goiter. So this is the goiter, the thyroid gland that was actually dissected out of an individual's neck, and you can see how large it is to where it is taking up primarily the majority of the neck portion. So this is what is the lack of iodine is stimulating, causing this gland to be much bigger. And just as we get goiter with humans, we get goiters with animals also. So this is actually a calf that was born. You could see here the goiter on its neck. So the cow was iodine deficient, and it resulted in the calf, as soon as it was born, it already had a goiter formed because the mother is going to keep the majority of the nutrients for itself. If there aren't enough nutrients in the environment for the mother, she will more likely result in reducing the livability or limit the nutrients to the fetus. So this fetus ended up developing a goiter before it was even born. So here we have another fetal calf that had congenital bovine goiter. So it was basically this goiter developed in utero due to the mother's lack of iodine or thyroxine, mainly the iodine. So since she had inadequate iodine, the fetus had inadequate, and you could see that it has the formation of a goiter already upon its birth. And when you do a necropsy on the calf, you could see, looking inside the neck, how large the thyroid gland is. The thyroid gland typically is very small, hard to see within the neck. So you could see upon its birth, usually they're not going to survive very long, and the thyroid gland was very enlarged in this calf. So next we're going to talk about magnesium. Magnesium is a divalent cation, meaning positively charged, and it is the most abundant cation found within nature. So yeah, it's found everywhere. And if you recall, it barely qualifies as a macro mineral because remember that most macro minerals are thought to be greater than 5 grams in a human body, where our magnesium is about 4.7. So it barely qualifies. It is the second most abundant intracellular cation, with the most abundant being potassium. And you have it's found between 60 and 65% in the bones, 27% in muscles, 6% in other tissues, and 1% in extracellular fluid. And It's bound to protein for emergency reservoir. So magnesium is probably one of the most critical minerals outside of calcium and phosphorus. It's probably the third most important. It is a cofactor for more than 300 enzymes. So it is needed for phosphorylation reactions. So if you don't have magnesium, we're not going to be able to have these phosphorylation reactions occur. So why is that? And that occurs because we need the magnesium here. So here we have our ATP. You can see our magnesium is stabilizing these phosphates. So here is one version where it's stabilizing the latter option, phosphate molecules. Here it is bound to all three. So this is actually stabilizing the negative charge on the phosphates, and that allows the ATP to better bind the enzymes that it needs. So it facilitates ATP binding to the enzymes by stabilizing those negatively charged phosphate groups. In addition to it being required for more than 300 enzymes, so with this, all of our kinases and phosphatases are going to be utilizing magnesium. So without magnesium, these enzymes are unable to work because we can't stabilize that ATP. And it's not just ATP, also GTP. Magnesium is also required for. In addition to that, we need it for our nucleic acid and protein synthesis. And it's also critical for our second messenger system. So typically when we're thinking about second messengers, we are thinking of calcium's role in that, but magnesium is also pretty critical in maintaining the second messenger signaling pathway. So here's a schematic showing how magnesium functions in its second messenger cascade. So here you can see if we have a hormone bind to its receptor, we need the magnesium here, so we have magnesium GTP to activate the G protein. We need it here for the adenylate cyclase. We need it for protein kinase A over here. We also need it to, if you remember our IP3, our signaling that we mentioned when we were talking about phospholipids in the lipid unit. So you can see we need magnesium for this to properly happen. We need magnesium to dephosphorylate our IP3, and this is important to get the signaled endoplasmic reticulum to release the calcium. We also need it for our protein kinase C over here. So you can see without magnesium, we're actually going to significantly reduce the amount of second messenger signaling that is occurring. So how do we absorb magnesium? Well, in non-ruminants, it's absorbed throughout the entire intestine. So from the duodenum all the way to the colon or the large intestine. But the maximum amount, the majority of it is absorbed within the distal jejunum and the proximal ileum. So those are the primary places that it's going to be absorbed. And we have that it requires a transporter when at low concentrations. So as this is a pretty common theme, low concentrations, we need a transporter. High concentrations, it's able to passively diffuse. There's not much known about the transporter related to magnesium. It has shown that it is somewhat dependent on vitamin D. So if you have inadequate vitamin D, you're not going to be able to absorb as much magnesium. So it's possible that vitamin D may be controlling magnesium absorption, similar to how it controls calcium and phosphorus absorption. So when we have really high levels, it's going to passively diffuse. Ruminants are able to absorb it from the rumen, and you can see here that it's not very available. So we're not very efficient at absorbing magnesium, so only about 30 to 50% of it can be absorbed. When we go to excrete magnesium, it's primarily excreted within the urine. However, it can be excreted in the feces via the enterohepatic circulation. So this is going with bile. When we release bile out of the gallbladder, that's how we can get rid of it. If we have excess that we have animals that are lactating, we can throw it in the milk, and we can also have our laying hens put it in the eggshell to get rid of excess levels of magnesium. So our deficiency in humans is typically associated with protein malnutrition. So one of the take-home messages with this class is really very rarely are we going to have in a free-living animal a one nutrient deficiency. So magnesium is high in meat. So we're low in protein, we have inadequate protein, we're more prone to have magnesium deficiency. If you have some renal disorder or endocrine disorder, you're more likely to get magnesium deficiency. Or if you have excessive vomiting or diarrhea, and this is more than what you would get with the flu, you don't have to worry about getting signs of magnesium deficiency if you just have flu for 2 or 3 days. So this is excessive vomiting, diarrhea, so more along the cases of, having Ebola or something, uncontrollable for prolonged periods of time. The symptoms of magnesium deficiency is going to be tetany. So this is basically when you have all your muscles contract at one time. It becomes it's very painful because basically you end up crushing a lot of your bones because both your for instance, your biceps and triceps are contracting at the same time. And if you know anything about muscle physiology, they're not meant to both be contracting, and eventually it would give way to your bones breaking. In addition, you can have some hallucinations that occur. So hopefully you observe the hallucinations before you get the symptoms of the tetany. Magnesium has been shown that it can protect against heart disease and hypertension. So it's not just sodium and chloride and potassium that can affect heart disease. Magnesium is also associated with that. So magnesium deficiency in ruminants is not all that uncommon. So with ruminants, it's what we refer to as grass tetany. And it's rather common. So this occurs when they are on a pasture or range that has low levels of magnesium or high levels of potassium. So our potassium and magnesium are antagonists. So if we have high potassium, it's going to compete and we're going to reduce or cause magnesium deficiency. So even if we have adequate magnesium but high potassium, we're going to get grass tetany. And this is very prevalent in the spring. So you'll see a lot of animals have grass tetany when you have the grasses regrowing after the winter season. They have a lot more rain and big spurts, so you're going to have much higher potassium in those forages early in the spring rather than in the later growing season, late summer. So here on the lower right-hand side, we have a cow that has breast tetany. So you can see here it's in a lot of pain. It's unable to move because all of its muscles are contracting at once. It's a very painful thing for the animals to endure. In poultry, we have observed magnesium deficiency. They typically get irritable, and you do get cases of tetany. But in poultry, we've mainly observed it when we fed experimental purified diets, where we are giving the single amino acids, where we're giving glucose and the specific vitamins and minerals, and it's very powdered, very unpalatable. But that's how we have discovered what magnesium deficiency looks like in poultry. It's not very reasonable. You're not really going to encounter magnesium deficiency in poultry. It's going to be more of a general malnutrition, or the mineral mix was left out of the diet. Our magnesium toxicity, you're not really going to encounter magnesium toxicity from a natural diet in people. Usually you're going to observe it from individuals taking high levels of supplements. With that, symptoms include osmotic diarrhea, nausea, cramping. You get metabolic alkalosis, hyperkalemia, and eventually, if you go on for a prolonged period of time, you can have cardiac arrest or heart attack. Cats, they can have increased urinary tract infections, and this is because of the stuvate formation. So you have magnesium, ammonia, and phosphate stones that form within the kidney, and it blocks the ureters and going to result in urinary tract infections. So here we have some pictures of our stuvate kidney stones. So here you can see this is a cross section of the kidney, and in that center part, the medulla part, that is loaded with stuvate kidney stones. So it completely blocks the ureters, so therefore urine can't flow, and this leads to kidney failure. So here you can see these kidney stones that have developed within this cat that was given high levels of magnesium. Our next mineral that we're going to discuss is zinc. So zinc is also a critical cofactor, and it's a cofactor for more than 100 enzymes. And the majority of these enzymes are metalloenzymes, so these are enzymes with a metal ion attached. It is found in the highest concentration in our bone, followed by, for our male species, the prostate, and then the eyes. However, our largest pool, the amount per organ, is our muscles. So our muscles have the greatest overall amount of zinc, but our bones have the highest concentration. So the two important functions of zinc is primarily, number one, it is a cofactor for these enzymes. And so it is binding the histidine and cysteine residues within these enzymes. And typically the zinc is doing this at the active site. So this is where it is stabilizing and exposing the active site of the enzyme, allowing the reaction to occur. When you have a deficiency in zinc, though, these enzymes have priority for zinc. So you will rarely see a decrease in the functionality of these enzymes because they get priority. If there's limited zinc, these 100 enzymes get first crack at the zinc, and then you're going to have lesser functions of the body are lower on the totem pole. Some of the enzymes that require zinc are found here. So we have pyruvate dehydrogenase, which is needed to convert pyruvate to acetyl-CoA, carbonic anhydrase, which is required for the carbon dioxide exchange, fructose 1,6-bisphosphatase, lactate dehydrogenase, carboxypeptidase, reverse transcriptase, DNA polymerase, superoxide dismutase, and various proteases and peptidases. So those are just a few of the 100 enzymes that it is involved in. The other main function of zinc is it forms zinc fingers, which is primarily also how it's working as a cofactor for these enzymes. But zinc fingers are critical for having proper DNA binding proteins. So by having these zinc fingers form on the various transcription factors, so this is in our transcription factors. So it forms these zinc fingers, and that is what's allowing these transcription factors to bind the start region on the DNA. So if you have inadequate amount of zinc, you're going to have a reduced amount of DNA transcription that's occurring. And it's also working here by binding the histidine and the cysteine within the transcription factors. So the absorption and metabolism of zinc. Zinc is primarily absorbed in the duodenum, but it can be absorbed throughout the whole small intestine. So also from the jejunum and the ileum, but it's primarily in the duodenum. And it's believed that the absorption is also similar to copper, so it's utilizing metallothionein to absorb, and this metallothionein is acting as a reservoir for both zinc and copper when we have excess intake. So because zinc is critical for all these cofactors, we don't want to just completely disregard it and excess be excreted into with our feces, that we actually have our cells absorb it and hold on to it through our metallothionein. So upon absorption, the enterocyte can either store it or it is involved in the cell metabolism. And similar with our magnesium, our zinc absorption is pretty low, so it ranges between 15 and 60%. And this range is so great because we have this it all depends. If we consume lots of zinc, we're going to actually be less efficient at absorbing it, where if we're deficient in zinc, we're going to be more efficient at absorbing it. So that's why the range is so great, between 15 and 60%. We can conserve it by reabsorbing it through the enteropancreatic circulation. And when the zinc is transported to leaves the enterocyte, it transports into the liver. It binds both serum albumin and transferrin. So our transferrin can bind and transport our iron, and it can transport our zinc. And then it's stored within the liver. Our liver can export our zinc using metallothionein and albumin as a carrier. So zinc deficiency is not all that uncommon. You see it a lot in individuals that consume diets low in animal protein and high in legumes, such as soybeans, so high in beans. So our soybeans have that nasty little molecule called phytate. If you remember from previous lecture, that phytate is an inositol ring that has phosphates on each of the 6 carbons, and those phosphate groups are able to bind our various minerals such as zinc, iron, calcium, magnesium. You can also induce a zinc deficiency by alcoholism, if you have Crohn's disease, and if you need intravenous diet, so if you, for whatever reason, unable to eat, you're getting all your nutrients through an IV, and they fail to give you the proper bag, and it does not have zinc in it, you can get zinc deficiency. This is another one of those fun antagonisms. So if you take high levels of calcium, so many, especially older women, are concerned about osteoporosis, and they know it is associated with the low levels of calcium within their diet. So they will take specifically high calcium supplements. And these high calcium supplements that they're taking for their osteoporosis actually can interfere with their zinc absorption, so you inadvertently get a zinc deficiency, one of those fun interactions or antagonisms that occurs. So during this, symptoms include that you'll get a rash on your face, groin and hands, you will get impaired immune function, altered GI function, you have impaired central nervous system and brain function. You disturb your thyroid function and metabolic rate, and you also have very slow wound healing. And a lot of this is all because of the zinc fingers that zinc makes through the various proteins, which make them more functional proteins. So here you can see the rashes on this individual that has zinc deficiency. So one of the common themes that you should see here is that it appears as though skin has the lowest priority compared to some of the other organs. So one of the main symptoms of these various deficiencies always results in some sort of rash. So then it's up to the physician to determine what is causing that particular rash. So here you can see on this individual, we have rash on the hands and feet. Here's another picture of an individual, then they have a zinc deficiency and you get this rash on the face. Now, similar to humans, our animals are also susceptible to zinc deficiency because of the amount of phytates within the diet. So, and it also depends on the source of zinc. So this is where a lot of poultry and swine producers, they may opt to utilize those organic minerals to where it's zinc conjugated to amino acids, which has greater bioavailability because it's those amino acids are protecting the zinc from being bound by the phytic acid or the phytates. So when you encounter zinc deficiency in animals, it's typically symptoms include poor growth, dermatitis, so you get a rash again, poor feathering, and then you'll see, especially in our chickens, they'll have a pale comb. So here's a pig that has zinc deficiency, so they have very rough, dry skin, and their hair is very brittle. So this is referred to as parakeratosis within the pigs, and this is all due to a deficiency in zinc. So here we have another picture of swine with zinc deficiency. So you can see the parakeratosis around the hoof region. And if you have the hens for chickens that have low levels of zinc, they're not going to put adequate amounts of zinc within the egg that is necessary for the chick to develop properly. So you get deformed chick embryos. So here you can see on the right-hand side, this chick, this embryo has not developed any wings. It does not have legs develop. And therefore, this embryo is not viable, and it will end up dying. If it does survive, it's going to have decreased hatchability, and it's not going to survive very long past hatching. So here we have a chick. It has very poor feathering due to zinc deficiency, and it also has a deformed hock joint here. That's known as swollen hock syndrome, and it basically results in abnormal thickening of the long bones because even though when we think of bones, we're thinking calcium and phosphorus, it is the highest concentration of zinc, so we're going to be breaking down some of those our bones to free up the zinc that we need, because our bones, similarly to the skin, have a lower priority for these minerals than other enzymes. So therefore, we get this abnormal thickening of the long bones. Here's another picture showing the parakeratosis on the breast and the leg of a deficient chicken. So you have this breast here, we have parakeratosis, and on the hocks, you can also see this rash that's developing, poor feathering. So that is all due to our zinc deficiency. So calves are also subjected to zinc deficiency. So this one in particular has parakeratosis on its legs over here and also around by its hooves. And if you were to look at this calf, you would see that in person, it's hard to tell from the picture, but it has very poor coat condition. So just remember the extremities, your skin, your bones, the coat, hair, it has the lowest priority for the majority of these nutrients. So therefore, they are the ones that are compromised and where we see the symptoms of these deficiencies.
So the next and final mineral that we're going to be discussing is manganese, which is abbreviated as Mn. And manganese is found within the human body at about 20 milligrams. So this isn't a standard 70 kilogram person. Within the cells, it's primarily located within the mitochondria, but it is also stored in bones. So we can store it in bones, but primarily in the majority of cells, it's going to be found in the mitochondria. It has 11 different oxidation states, but because of that, it is known as a transition ion. But for the most part, it exists either in the +2 or +3, similar to iron, for the majority of reactions in animals. It activates many enzymes involved in fatty acid metabolism and protein synthesis. It's also required for normal thyroid function, so we need this for the formation of thyroxine, which, if you recall, we just talked about with our iodine. So manganese is really poorly absorbed. It's only about 2 to 15% is absorbed, and that's due to the high amounts of phytates, tannins, and all the mineral antagonisms that are involved. So, as I mentioned, it has 11 different oxidation states, so therefore it can interact with many different minerals, and it has a lower affinity for many of the transporters. So it has a lower priority. All of these are contributing. So deficiencies in humans is actually pretty rare. So here we can see some pictures with the pigs with our manganese deficiency. Here you can see our pig on top on the left-hand side. It is deficient in manganese, and you can see how much fatter it is compared to the control pig on the bottom, which is much more lean. In addition, on the right-hand side, we have this piglet that has this tripod stance that it's requiring to balance itself and try and right itself. And finally, the manganese deficiency in poultry. Here we have a chick that has porosis, that slip tendon in its hock, so it's unable to walk adequately. And that's really where we discovered, it was primarily in the chicken where we found these clinical signs of manganese deficiency. In addition, on the right-hand side, we have a microscope, increase of magnification, looking at the pores of the eggshell. So you can see up on the top portion, this is our manganese deficient egg. So it came from a hen that was deficient. You can see how much greater these knobs are and that there's more space. So we have bigger knobs with more space. So that's really what's resulting in our decreased eggshell quality compared to the control down here. The knobs are smaller, resulting in smaller pores, stronger eggs.